introduce Grace Weber and Elliot LaTray. I am Rose Pistic. I am the McLaughlin Research Institute Animal Facility Director. And they waltzed into my animal world this summer and learned how to connect to all of our little citizens and help me with a very big project that I had going on, figuring out what is, for behavioral testing, the optimal conditions to create a calm, well-centered mouse. An anxious mouse is not going to participate in a cognition test. So any of our Alzheimer's studies, where we're trying to test whether or not the things we're doing are making a difference as far as learning, memory recall, an anxious mouse is not going to participate. And in your brochures, if you're in your program, if you did read the introduction, we have a collaboration with Dr. Miranda Orr, who is a Montana native, and she's at Wake Forest University. And she does lean on us as a facility not only to generate the mice that she needs to test, but she is starting to utilize our behavior capacity. And we did a very well executed study last summer that another University of Providence student, Sarah Terham, was helpful on, and another Great Falls native, Wyatt Walters, was helpful on. And we did get some data back that maybe our behavioral suite has some anxiety-inducing factors that we need to try to figure out. So again, they came in and stepped up for that. And I do want to say, parents, that it was their idea to come in and test in the evenings, because that seemed like the less anxious time for a mouse to be tested, since that is their active cycle. So I will let Grace and Elliot show you what they present this summer. Oh, I can help you with that.
Okay, like Rose was saying, for this summer, Elliot and I were troubleshooting anxiety screening tests.
For a little background on why we care about the anxiety of mice is mice and humans learn similarly, and their ability to learn and remember is very similar. So if we can understand what makes mice anxious for these tests, it'll give us better results for, just give us better results in general for the test. Quote we put on there was, learning is an adaptive process that's essential for day-to-day functioning and overall survival of organisms.
So why were we doing these anxiety screens? Anxious mice, like Elliot said, will not perform in these cognitive tests well. So we are trying to create conditions where the mouse is calm and ready to learn and not stressed. These tests pre-screen mice for anxiety so they can perform in future cognitive tests. For our tests, we did two pre-screening anxiety tests. I myself did the dark light exploration test, and Elliot chose the open field test. The two questions that we were trying to answer were, can we score anxiety with the dark light test or the open field test? And are there any strain-specific behaviors that come with these tests? We chose two groups, one group with a known anxious mice, and we induced anxiety into these mice by creating a stressful environment for them, and one group of anxiety-controlled mice. We followed the guide protocol to help make these mice the least stressed and anxious as we could. We took a video of the test and used the AnyMaze tracking system to record their behavior. We used two strains of mice for these tests with different sexes and about relatively the same age, and the test mice were at about the age of 5 to 7 weeks. The animals we used were the B6 MAPT-null mice, the FEB MAPT-null, the FEB B6F1 MAPT-null, and the ICR wild type. And for our test, we had a population size of 12 for the anxiety group and 56 for the control group.
All right. For the two groups, we had the anxiety-induced and the control group. For the anxiety-induced, we tried to make them as anxious as possible. So we did things like house them alone, house them on top shelves with more light than the as opposed to the control group, which was housed in the middle rows with about 30 lux for the more anxious mice, 25 lux away for the control group. We handled them by the tail, didn't acclimate them to the room. During the test, we tried to make it a little more noisy by walking around, talking, moving, all that sort of stuff. They were not handled prior to testing, and they were tested during their usual sleep cycle, which is at 8 a.m. And during the test, we had a light shining on, which was about 250 lux light. For the control group, they were housed in groups, shelved in the middle row. Instead of being handled by the tail, they were handled in either a tunnel or a cage. And then, in their box, they had two forms of enrichment, which were a tunnel or a cage to make them feel more safe and have a little place to hide if they needed to, as opposed to the anxiety mice, which had not so comforting things like a chew stick or a cage swing. And they were housed in the behavior room at least a week prior to testing to make them more comfortable with the room. They had a quiet testing area, and they were handled about at least once a day for at least a week. And they were tested during their wake cycle at 8 p.m., and their testing was done under lower light conditions. Then on the left is the open field testing box, which is what I did. On the right is the light dark test box, which is one Grace did. And then in the middle, you can see on the top is a picture of the control mice housing, and the bottom is the anxious mice housing, which is those boxes on the top. And then this is what the housing looked like for the mice. For the control group, they had their little tunnels, and then housing groups. Then these were some of the mice we used. On the end is the B6 Maptinol, the middle is the B6 FEB1, FEBF1, and then the ICR wild type on the end.
All right, so for my light dart exploration test, some background on this test. It was developed by Crawley and Goodwin as a precursor for the elevated plus maze, as well as other cognitive tests that will follow the pre-screening anxiety test. In this test, it examines anxiety-like behavior in rodents. Mice exhibiting higher levels of anxiogenic-like behavior will make fewer transitions between the brightly illuminated open area and the dark enclosed compartment. Time in the dark will reciprocate time in the light as a measure of anxiogenic-like behavior as well in this test. For this test, they were exposed to a novel environment, the dark side that is protected and the light unprotected side. It also got measurements for anxiety and stress using their distance traveled in the light compartment, their percent time spent in the light compartment, as well as their number of transitions. For the materials that we used for this test, a square plexiglass chamber about 40 by 40 by 20 centimeters, and that was divided into equal compartments, at one side having transparent walls on the light side and black walls with black hinged top on the dark side, as well as gray floors. We used a web camera, the AnyMaze tracking system as well, and the 70% ethanol or Tetrol, genetically defined mice, and an ALAC accredited behavior suite. For the procedure, we first would acclimate the mice to the behavior suite room, set indirect lighting about 8 lux for the light chamber. We would singly place the mouse in the center of the chamber in the light side facing away from the dark chamber. We would set a timer for 10 minutes and video the tests for analysis later. After those 10 minutes were done, we would take the mouse out and put it back in its home cage and sanitize with 70% ethanol between the tests and then repeat for the next mice. Some of the results that we got. So for our first question was, can we score anxiety with the dark light test? We first grouped the ICR wild type anxious versus the ICR wild type control group, the non-anxious mice, and we found there to be significant difference between the ICR anxious group and the ICR control, where the ICR anxious mice traveled less, spent less time in the light chamber, and made less transitions overall. We then looked at another group with the FEB Maptinol mice that were anxious and the FEB Maptinol that were the control group. And they showed there to be no significance, and their anxious mice had the same data as the FEB control mice did. This led us to ask another question. Are there strain-specific behaviors for this test? So we first looked at the groups, the anxious group together. So the FEB Matinol anxious versus the ICR wild type anxious. And as you can see, they had significant differences in their data, where the ICR traveled less, spent less time in the light chamber, and made fewer transitions. This led us to think that the FEB Matinol mice were more of an approach exploratory behavior than the ICR wild type, who had more of an avoidance behavior to these tests. We then looked at all these strains as a whole and thought that these identical genotypes, the different background strains mice, exhibited different behaviors as seen on the graph. This test is sensitive enough to pick up the differences between these strains. We then went on to control for one of the variables that caused anxiety, and we chose to test mice at 8 a.m. versus testing the control at 8 p.m., which were tested at 8 p.m. For the B6 Matinol, they showed no significant difference. We then tested the ICR wild type at 8 a.m. and then the ICR wild type at 8 p.m. And they too showed no significance in their time. And then for the final group tested at 8 a.m. were the FEB B6F1 and the FEB B6F1 control tested at 8 p.m. and they too showed overall no significance. And this led us to believe that it doesn't really matter what time these mice were tested. It did not seem to affect their anxiety levels. With these strains, the time of testing did not seem to matter. We then controlled for another variable, which was the experimenter. Throughout the weeks, I was handling my mice and then testing them by myself, which were the control group, where the ICR wild type tested by Elliot were handled by me, but then tested by Elliot. And this showed there to be a significant difference between the mice tested by Elliot and the mice that were tested by myself. It suggests that we should ensure that the handler and the tester are the same person to reduce the anxiety levels of these mice. So for some discussion, can we score anxiety with the dark light test? With the ICR, there showed to be a significant difference, and with the FEB mice, there showed to be no significance at all. And we believe that every line should be screened for anxiety before testing by having an anxiety-induced group and a control group. And so for the second question, are there any strain-specific behaviors for this test? We tested many groups, and they showed to be either significant or non-significant between the times tested and the handler. And we also saw that some of these mice had an avoidance behavior to the test, and some of them had more of an approach behavior to the test by traveling more, spending more time with light, or making more transitions overall. And so we believe that it is significant strain. We have significant strain-specific behaviors in these tests. And how is this useful? It is useful because we are able to screen for anxiety-like behavior, anxiogenic-like behavior with the dark light tests. We are able to understand the different strains of the behavior, and this test is an ability to have a first step screen to figure out if these mice are ready for further behavior testing. Some errors we had in this test were that the AnyMaze program was not perfect in its scoring, and to fix that, I believe we should make sure that all those errors are troubleshooted before we actually start doing these tests to have perfect scoring. And then another error we had was the population size for the anxiety group was too small, with a population size of about 6, compared to the control group who had 20 plus mice in it. And so we should gather a good population size for these mice of about 5 plus for each strain in the group. And then the final error we had in this test was that the B6 mice were not tested with an anxiety group. And for further testing, we should prepare a test where all strains have a control and anxiety group and anxious group. And so for this test, like I said, we didn't have an anxious group for the B6, so we were able to compare their non-anxious strain to the anxious side of things. And for future studies, we should change anxiety variables to see if there is a factor that affects other mice more than others. And so like changing the time of the testing, maybe changing the light, changing the handler, like any of those factors that could cause anxiety. We should have a larger population size that is consistent throughout the whole study. and we should make sure that the NMAs will not have any errors during the analysis readings.
For my part of the summer project, I was utilizing the open field exploration test as an anxiety screen, and a little background on it. It was made by Calvin S. Hall, and it evaluates curiosity, will to explore, and the general anxiety levels and locomotor activities of the mice. It can be measured using the test with things like sticking to the wall or showing behavior known as stigmotaxis. And that is usually a sign of anxiety, as opposed to a mouse that explores more and is in the center a lot, and that's usually seen as will to explore and less anxious mouse. Materials for my test, I use a 40 by 40 by 20 centimeter box with opaque walls so they can't see visual cues outside their testing area. An ALAC accredited behavioral suite, genetically defined mice to use in the experiment, web camera and image tracking system, infrared light for testing in the dark, then 70% ethanol and paper towels to clean after each test. The procedure, first we acclimate the mouse to the room for at least an hour in advance. Then after the acclimation period, we take the mouse out, bring it over to the testing area, hit record on the computer, start the timer for 5 minutes, and put the mouse in the middle of the box. Then once the test begins, we try to exit the room as quietly as possible. And then we wait till the timer goes off, and when it does, we go back in, stop the recording, stop the timer, and take the mouse out. And finally, clean the box accordingly and prepare for the next test. We have results from, we use an image tracking system to measure a few things like the average distance traveled of the mouse, the average speed of the mouse, and then the average time spent in the corner and center zones. For our first question, I also did the community score anxiety using the open field exploration test, and I did the ICR wild type anxious versus the ICR wild type control. And for these ones, it reached no significance. Then I went on to do the FEB matinal anxious versus FEB matinal control, and once again, no significance. And after testing within their own strains, we want to expand the question and directly compare two groups of anxious mice from each strain and two groups of control mice from each strain. So we did the ICR wild type anxious versus the FEB matinal anxious, and no significance here either. Then did the FEB matinal control versus the ICR wild type control, and no significance. So in addition to those things, we'd like to explore other factors that may make the mice anxious, like time of day testing, like testing during their normal sleep schedule, which is 8 a.m., or their normal wake cycle, which is at 8 p.m., and also tested by an unfamiliar or a familiar handler. So first off, I did the ICR wild type done at 8 a.m. versus the ICR wild type done at the control done at 8 p.m., and no major differences, so no significance. Then I did the FEB-V6 control versus the FEB-V6 8 a.m., and no significance here either. So after that, I did the ICR wild type tested by Grace versus the ICR wild type tested by me, and once again, there's no significance. So conclusions for these is there weren't any major differences between the two strains, and in addition, there didn't seem to be a difference between the time of day tested, so or the handler, so neither of those seem to make much of a difference when testing. And then my next question is, are there any strain-specific behaviors for this test? So for the FEB matinal control versus the V6 matinal control, once again, there's no major differences, so no significance for this data. Then for the FEB V6 control versus the V6 control, I actually I had 2 out of the 4 points that were significant, and then one was close to significant, so about 3 out of 4 were significant, so it would probably be good to do a little more testing on these mice and see if anything comes of it. And then after that, I did the FEB-V6 control, the FEB control, and there was no significance for this data. Discussion, can we score anxiety with open field exploration tests? And we came to the conclusion that no, it would not be a good idea because there were no major differences or significance in the data. And for the second question, are there any strain-specific behaviors? No, none that we saw or got from the data. How is this useful? It's useful to know an animal's anxiety levels before putting them into a test, because if they're too anxious, they either won't be able to perform very well or they won't be able to perform at all. So understanding signs of stress and anxiety before putting them in a further test is good to know, so then you get the most accurate results you can. And therefore, if you're collecting and analyzing the data from my test, we saw that the open field exploration test would not be the best option for pre-screening mice for anxiety before tests. And then acknowledgements, we'd like to thank Rose Pitstick, Mike Kavanaugh, Katie Evans, Renee Rejo-Pera, Derek Silvius, and the private donors for helping and funding our project.
If you have any questions, they are ready to field any and all questions.
So congratulations, those are absolutely fantastic presentations. I would have thought you were graduate students or students or something. They're great. I just had a curiosity if you know anything about this. I was wondering, besides the behavioral aspects, if you know anything about anxiety and eating or drinking in mice. I was, or if it would, if you saw, do the mice appear to be anxious just by looking at them, besides the tests and whether or not?
So we saw a lot of some of the strains like grind in their food and stuff like that, and we handled some of those mice for 2 weeks and they still were not happy with us by the end of the 2-week handling. Like they were still trying to bite us, and they bit Elliot, and they were fighting with each other. They just weren't nice. And whereas some of the other ones, like the ICR that were handled for a week, they were so happy to see us. Like they were just the nicest mice ever, and they didn't grind their food or do anything like that. Like that was one of the major things that we saw, and that was the handling. Like some of them just didn't like to be handled like as much as we tried, so that may be something that caused them more anxiety and yeah.
So most of your strains are inbred strains, so they should be genetically homogeneous, but the ICRs are outbred. Would you say that your data suggests that there's a genetic predisposition or effect on anxiety in what you've looked at?
So the ICR seem to have, for my test, they seem to be more affected by the anxiety. Like they, at the beginning, like see the anxious group were anxious and the non-anxious group were not anxious, whereas like the FBB mice and the B6 mice and the FBB B6F1 mice didn't show as like drastic significances between each other, so like, that could possibly.
That actually surprises me because I would expect the outbred group to have more variability.
Yeah. So that was, the ICR seemed to have, and even when we controlled for the variable like the handler, the ICR were the only ones that had shown significance in that too. So.
When you were making your anxiety group, there was one sense that I feel like might have been left out, and a big one with animals is their olfactory stimulation. Their sense of smell.
Okay.
And you didn't do any toxic sense of smells or anything like that with the anxiety group, correct?
We did not. We just housed them on that top shelf in the main room, and they were housed alone and just kind of together, just like the 6 that were being tested together. Yeah, we didn't take any of that.
So is there anything you can think of when you were actually running the tests that might have induced anxiety related to their sense of smell when you're running your tests?
For the control group and stuff like that?
For both groups, yeah.
So we did them in the testing room. So honestly, we put them. The anxious ones could have had something like that because they were brought into the room and like new to the smell of that room. But the control group were actually housed in that room, so they were used to the familiarity of that room. So
in between mice, when you were cleaning the cages and you were using the ethanol, was it did you let the cages air out after
Yeah,
that?
so it was about 3 to 4 minutes in between each test, and it was, yeah, we dried it and made sure every place was dry, and then even at the beginning, when we were starting the testing, like the first mouse of the day, we even like ethanol it, so the ethanol was the same, relatively the same for every mouse, so they smelled, yeah.
Great.
Do you think that the handler could be an olfactory influence?
That could have been when Elliot handled the mice, or
I know, I was wondering that, but then you guys were, it looked like it was the same, right?
Yeah, so the only,
One but not the other.
yeah, the ICR mice were the only ones that had the significance on mine, but his showed no significance really across all of his data, but that could have been something also.
So, Elliot, you went through some of your data pretty quickly, and I was wondering if you could go back and talk about I think there were like 3 or 4 tests where you said you did see that there was a statistically significant difference.
Oh, yeah.
Maybe just.
Oh, I think it was this one right here. Yeah. Oh wait, so what would you want?
So, what is your takeaway from that data in terms of significance in difference in behavior?
Okay, so I think this was the second question. Yeah. So for the strain-specific behaviors, I believe. I think it was just. Kind of stumped me a little bit.
Well, so like the average distance, for example. I guess one question is maybe you could talk about what does that mean, if the p-value is 0.03 versus 0.6.
So yeah, if it's under or over the 0.5, or 5%, I guess. I guess that would be significant because I guess if it's strain-specific behaviors for the test, I guess they both act differently and such. And then for the p-value, I think it's if it's under 0.05, that's significant because I don't know, yeah. It was,
I didn't mean to stump you. I mean, I think it's kind of, it's sort of almost kind of the standard. It's empirical in a way, but it can be tricky to interpret whether something's significant or not. And, sometimes if you repeat it enough times, there could be a really small difference and it emerges, but it takes a lot of measurements to see it reliably.
So some of your studies you just need to might throw more stuff at it, and you might parse some things out because there were some trends in your data. We don't have to go over every graph of it, but if you just plain old add more mice to it, you might actually reach significance just in number.
But you've also done multiple tests, and sometimes you need to correct for that. Because a p-value of 0.05 is basically saying you're looking for less than a 1 in 20 chance of it happening by chance. But the more times you test the data, the more likely you are to hit that 1 in 20, right? If you do it 20 times, you probably get it once, even though it's not significant. So if you do multiple testing, sometimes you have to do a correction and you use a more stringent p-value. So I don't know with the data that you tested or how you tested if that would be necessary or not.
This graph is really nice though, because it kind of makes sense when you look at the first two, that the average distance traveled by the blue mice is less, and they're also going slower.
And
So
they're hanging out in the corner more.
yeah, so it's really consistent that if you're moving slower, you're probably going to cover less distance. And if you're hanging out in the corner,
Are the last two labeled correctly? Because it says they're the same thing, but the data's different.
Time spent in the center.
Yes.
Oh, so I just can't read.
I don't get that first two. It's like reread it.
Yeah,
Yeah, a question for you about how you defined center versus corner for the open field. Do you remember how you did that?
Oh, yeah, with the anybody's tracking system, there was like, you could kind of cut the box up into like a grid, and then we put like one box, checked like one box each corner, and then when the mouse entered that zone, it became as like being in the center zone, or the corner zone, then we put, I think, like, I don't know, we kind of selected an area in the middle, and then they entered that area.
So was there are they either in the corner or in the center, or is there mid-ground there
There is
too?
mid-ground,
Okay.
Yeah, so I think it was like a 4x4, so it was like 16, so we had like 4 squares, 4 in the corner, and then 4 in the center, and then I think there was 2 for like the sides.
Okay.
Also.
Should you track the sides, like if they're right against the edge? Because earlier you said that that's an anxiety thing, is being on the edge, maybe not necessarily in the corner, but just on the edge.
Oh, yeah. Fair point, yeah.
It might be interesting to compare just the corners to all the.
So the reason we chose these ones were because we have been looking at previous tests that were done on the open field, and these were some of the data points that they collected on their tests. And then the NEA's tracking system, when they were setting up the open field, they had selected just the corners and the center, which was coherent with what the data we were seeing from other tests.
One of the things I thought was interesting was there might be differences in handlers. I remember when I did some mouse experiments the very first time in my career, I think I caused every mouse to get very anxious. And so I used to go to the person that was Rose's equivalent, and she could calm them down, you know, so that I could get blood or whatever I was trying to do. But I had this feeling that they saw me coming, and they would get anxious just by the fact that I'm approaching the cage.
And that's why we chose to make that one of the variables, as if I handled all my mice for a week and then Elliot ended up doing that test on them, or vice versa, then we could see if that made a huge difference, and it was significant in my data at least. And handling was definitely a big part of handling those mice weeks before, like a couple weeks before they got tested, versus not handling them at all. Like like I said, by the end of that week or 2 weeks, most of the mice were nice enough to just like walk into your hands as you cooked them, and some of them were just not.
Yeah, I would love to share. One time I came in to see how everything was rolling, and I think I was in there to like consider maybe a different way to mount the camera, I don't know, but I had her put a test mouse in, and so she put one of her test mice in, and then I said, okay, we're done. And so she reached in her hand and scared the bejeebers out of the mouse. She just reached her hand into the box, and the mouse like just jumped, and she goes, oh, I'm sorry, and the mouse turned around and he's like, oh, we're all good, and just let her pick it up just like that. Just so calm. I was like, wow. All right, well, thank you all so much for coming.
Thank you.
There are pizza questions right out here.
I do want to say